Good afternoon, and thanks again for giving me this opportunity. So, I think when I in Izmir, Cape Town, first I met Steph and Pedro, and then I came to know about this datathon, and then it was really my pleasure to join datathon and to present some of my work I'm doing in Africa. So, uh, so I will be talking about microbiome data analysis in terms of environmental sustainability, and I will show some data, what we do exactly at AGC, which is African Genome Center at UM6P in Morocco, and at the same time, some of the key project which is ongoing. So first, as we all know, if we take a gram of soil that contains more than 50,000 of microbial species, and in terms of genes, it's more than 150 million unique or partial genes exist. So from a 1 gram of soil. And we also know that microbial communities in the soil are very important for ecosystem balance in terms of nutrient cycling, carbon sequestration, also carbon matter, organic matter decomposition. So that tells us that soil microbiome or microbes in the environment are so important, not only for the plants, also for other perspectives or the factors controlling or balancing in the ecosystem. This is the recently published paper in 2023, and AGC was part of that, entitled African Microbiome Matters, published in Nature Microbiome. So in this figure, you can see all these colorful dots are the microbiome datasets submitted or deposited from all over the world to NCBI. And if you look for African part, it's quite empty. There is not much. And this paper also mentioned that Africa has a wide area of land, but in terms of microbiome data set, we are still producing very low comparing to the developed countries. But African's ecosystems are one of the most genetically diverse ecosystem in the world. And also there is a huge diversity in connecting plant, animal, human populations in all over Africa. But because of the lack of microbiome research, the data is not enriched much. But they also say that African microbiome is unique and quite unexplored. So what proposal they are giving is to, in terms of strengthening the microbiome studies in Africa, the first one is to initiate a coordinated research network within the continent. They are also proposing to implement the policy interventions to develop the science and research planning in order to focus on African innovation, increasing capacity, and also increasing the number of publication or the outputs throughout the continent. And another thing, it is really important to develop the infrastructure in African continent, and it can be like there could be a different sequencing and microbiome center of excellence in different major parts of Africa in order to development the microbiome research or strengthen the microbiome research in Africa. So in order to capacity building, so what we can rephrase these words are we really need some pan-African effort in terms of genomics and bioinformatics. If we want to really overcome the barrier for microbiome research, then we should implement regional and continental based training and workshops, educational curriculum development, and also we should support the scientific entrepreneur development for the African's bioeconomy. So at AGC in UM6P in Morocco, we are really focusing in these two parts as a part of the training and also a research. So we also did a couple of short-term and residential training over the last 2 years. We are also doing internship, and of course, we are always welcome and open for the collaboration, not only in the continent, even outside of the continent. So here, this is an example of what AGC we did over the last 1.5 years. We organized 4 different workshops, and the first workshop was environmental microbiome. So what we really do, we advertise and then we select the candidates. And the first couple of them were only for the PhD students from Morocco. And we don't do only dry workshop or the training. This one was completely in-hand experience in the lab. So the students were in UM6P for 3 days. They were getting experience from DNA extraction, then quantification, purification, PCR, library preparation for sequencing. And once the run was done, we also trained them how to cure the data. I mean, basically, this was 16 days data and up to how you get done the analysis and get your data for publication. Second one was biogenome project. It was quite similar, but not specifically on the 16S. It was quite overall for library preparation and sequencing. And again, it was for 15 participants from all over Morocco. Then the third workshop we did last year, again, it was only from the students and researchers from UM6P, and it was very specific for bacterial diversity genomics. So we have sequenced 16 bacterial isolates using NGS method, and we analyzed the data, and now this is ready to publish, and it's going to come soon. The last one this year we did was quite interesting because it was completely different than the other three. It was whole genome sequencing of an endangered plant in Morocco. The plant was Acacia gamifera, and we selected 10 students and scientists from all over Africa, from 10 different countries. And surprisingly, when we advertised, we got like around 3,000 applications, but we were really forced to narrow down 10 to train them really in hand. And as I said, that we always did like publication-based training. And one of the key example is from our first and second workshop. Some of you might already have known these perspective articles published in Nature Genetics, and AGC is a part of that. So it was how we established this African Genomics and Bioinformatics program through a different regional workshop. And just to mention that, we have conducted 23 awareness and hands-in training workshop on genomics and bioinformatics in 5 African geographical locations and 11 African countries. And in Morocco, University of Mohammed VI, and especially AGC, played a key part for doing in-hand experience. And as I showed, first 2-1 was part of this. Also, South Africa, Nigeria, and Kenya, they played a very good role for conducting this kind of regional workshop. And this was the last 10 participants who were selected and participated the whole genome sequencing of acacia plant. They were from 10 different countries, and it was for 2 weeks long. And as we said, this is a residential training because they were here and they stayed. Their accommodation, food, and the training, everything was covered by AGC. And the plant we sequenced, as I said, is from the Fabaceae family, Acacia gamifera. This is one of the endangered red flag plant from Morocco and medicinally important. So we are now analyzing that data and waiting for some of the data from PacBio just to complete the analysis. So this is the plant we have sampled from one of our plant in experimental farm at UM6P campus. And sorry, continuing that, so as we said, we do conduct more workshop, and the next one we will be announcing in January, which will be we call quite extended. It's kind of internship, 16-week long training. And the reason we go for that long is we start from sampling, we complete the whole experiment, then analysis, then creating the data, drafting the paper, and we submit. That's why it's going to be quite longer. So that's like how we can actually, that was overall how we are actually exploring all for Africa to enrich our microbiome data in the data set. And I'm happy to say that this now is going to be the part of the datathon as well. So now I will just give few examples of what kind of research I am doing or AGC is doing related to microbiome. So one of the key project I am running in Sahara Desert is mapping microbial diversity in the southern Moroccan ecosystem for sustainable agriculture. So this project is basically funded by Fospo Bokra Foundation, is a part of the OCP group in Morocco. And at AGC, we are doing this conducting all this research in support of ASARI, which is another institute in the southern Morocco. And this is also a part of UM6B University. So southern Morocco, some of you may know, is quite dry. And this is a photo, from the dry river in southern part. So you can see how dry it is. And there is a huge agricultural challenges in this arid region, especially for water and salinity is a big problem. But still there are some plants growing there. So we are trying to explore this area because this is quite unexplored. There is no microbiome research and data available from this region. And we are expecting to find out or map this microbiome in that region in terms of crop productivity in fighting with saline or drought. So the objectives of this project was to explore the microbial community diversity, then to investigate and assemble the microbial community structure, how these factors are shaping the environment. Then the third one is analyzes the significant variations within the diverse microbiome in diverse microbial ecosystem. As when I'm saying that a diverse ecosystem is because I will show in next couple of slides how the ecosystems are quite diversified within like 500 kilometer square area. And then we really want to uncover different pathways and networks involving in microbe or plant-microbe interactions that could help us in enhancing plant resilience under this extreme environment. And the overall key objective is to map the diversity of microbial communities associated with drought resistance plant across the arid regions in southern Morocco. So this is our the study area in the southern part. If you see, comparing to the north part, is completely dry. So these are the 5 locations we have sampled. Those are quite really far, like almost 5 to up to 800 kilometers from each other. And this is some of the snaps for how we were doing the sampling in the desert. So now, as I'm saying, I say the different ecosystems. So we selected 5 different ecosystems. The first one was dunes, then Grara, Safka, lagoon, and river. And I will show you how they are really different. So let's talk about the dunes ecosystem. Dunes, we all know about dunes is like the sand dunes. So it is dominated by like moving sands. It's a very windy, erotic area, low organic carbon matter, and you barely find the plant there. And even though you find there is only 1 or 2 types of plant, they are surviving there. Then Grara is we called it low-lying basins because occasionally after the rain, it holds some water, if you can see in the figure here. But in the dry season, you will see maybe nothing there. So this is also another challenging environment for water scarcity. Then the third one was Safka. So it's mostly in the coastal part, but it's a highly deposition of the salt. So it's mostly halophytes plants are surviving. If you look at the arrows, you can see next to the water, there is a clear deposition, whitish part. They are like, they are the salt. And if you go there, you can really touch the layer of sand is like too thick. And the river, the fourth one is the river ecosystem. So if you look at this figure, top and the bottom one, this is the same river, 400 kilometer upstream and downstream. So 400 kilometer downstream, there are still water, and upstream there is no water, and there is completely different plant types are growing there. So This is again, it's the rivers depends on how which part you are, and depending on that, this salinity level and drought level are completely different, and also adapted plants are different. And the last ecosystem we sampled was coastal lagoons. So these are mostly wetland areas, waterlogged, saline, with frequently fluctuating salinity water are there. And it is very humid. Just to get an overall picture from high temperature and humidity, you can see here dunes are very dry and low humid comparing to the lagoon. And that's why I said that they're really diverse in terms of water salinity. So the sampling details, we sampled 5 different ecosystems, but 12 different sites, because some of the ecosystem we sampled more than one site, depending on the geographical distribution. Just an example is, as if you remember, the 400-kilometer upstream river and the downstream river was completely different. So we needed to sample 2 or 3 different sites for the river, and same thing for Grara ecosystem. So in total, we sampled, I think, 600 something, but there are only total 34 different plant types. 200, but these are the number of the samples for rhizosphere, root, and endosphere. We did 200 for each, and 28 for bulk sample. And then the total, as I said, 34 plant species type, and sorry, plant group distribution. So 24% were tree, 48% were shrub, and 27% were small vegetations. And here are the type of different plant groups um, according to the family and species, and those are highlighted, like let's say Tamariaceae, Chenopodiaceae, Asteraceae. These are families are common in different ecosystems. So it was very difficult to find the common plants in multiple ecosystems. So this is the part of the group. We went for sampling for more than a week, and thanks to our this mobile lab, it helped a lot, because we started sampling at very early morning at 5 a.m. and sometimes we came back midnight at 12, and that mobile lab really helped us to keep our sample alive. So the overall strategy from this sampling campaign and this project is, as I said, our main objective was just to collect from different parts of the plant, then processing for their physiochemical properties for the bulk soil and plants. We already did this part. Now we are doing 2 different main approach. Those are the 2 main part of this project. The one of the main part is the culture dependent approach. So it's like thousands of samples we are culturing, and we did not conclude this yet, because this project we just started 9 months back. And for the culture dependent approach, we are mostly now at the very beginning, we are doing a little bit of 16S and ITS, but not for all of them, because we really want to see how diverse and how the results goes. Then our main target is to go for the metagenomics, and we are also planning, like, to really, as our main target is to microbial mapping. So we will do the metagenomics, and at the same time, if the time persists for this project, if not, then in future downstream of our aim is we'll also do some functional analysis in terms of metatranscriptomics. And overall goal is to map the whole microbiome in that region, and at the same time we will try to look for some novel PGPR or PGPM candidate which can be used for the sustainable agriculture in the future. So this project is still ongoing, because we are just last week we processed the sample for sequencing, and we are waiting for some of our sequencing result. So there is another project I'm conducting is on argon. So we want to see how the distribution of argon and understanding if its connected microbiome functionality is associated. So you may know argan is a very important plant for Morocco, because this is the only place where argan grows, and this is very economically important in terms of oil and some other medicinal benefits. But this plant is also under potential threat. And uh, another thing is, according to UNESCO, I think around 2008 or somewhere, they declared it as a heritage. So in the part of Isawara region, so they have announced this is the Argon Heritage Region to protect this plant. And another thing to mention that this argon, if we look at this figure, that all over the place, if you look up of the photo, that is a huge mountainous area. These all the plants, green trees you are looking, see, you can see these all are the argon tree, and they are naturally growing there. It's not that artificially cultivated. And on the left side, see these 4 or 5 region, Agadir, Essaouira, some of the southern Rabat and Barkane in north. These are mostly naturally argon growing area in Morocco. And if you look a close view, that when this is the photo we took this summer, when it was like really fully fruited, and people are waiting until they ripe to collect the fruits. And if you look very closely, so these photo are the seeds of argan fruits. They are from different parts of Morocco, and they are really diverse. If you see the same time, within 1 week or 2 days difference, we collected the samples. In terms of shape and size, they are completely different. So the approach we are using for argan is like we sampled all over the Morocco. Then our 2 key approaches are metagenomic studies for microbial diversity mapping, because it's not has been done yet. Then we will also want to see the metagenomics for different parts of the microbiome in the plant, like for example, fruits, shoots, roots, and rhizosphere, because fruit is one of the most important part for argon for its oil production. And I'm not going in detail for the process because we already went through since morning. We are going to use this traditional high throughput sequencing system for DNA, and at the same time for functional studies, we also sample for RNA. We will see in future if we can do metatranscriptomics in order to find out the functional properties. And another thing we are doing with this, as we have sampled in different geographical location and also in a spatial distribution. So we really want to see how these microbes actually behaves at different altitude and at the same time at different spatiotemporal distribution. Are there any spatiotemporal distribution associated with these argon roots? Because if you look at the argon tree, this is really huge tree. And if you go for sampling, you will see sometimes to collect the roots, we really need to follow the roots like almost a 1 meter far. So, and then we did sampling in a different distribution. So we want to see how they are really actually distributed in a spatial distribution. So our main goal is to address the factors associated with adaptation of various geographical location. Sorry, and understanding the role of microbiota associated with argon. And we want to see the association between genetic diversity and argon microbiome in order to provide insight into the functional role of if is there any core microbial taxa for promoting the growth and nutrient acquisition or stress tolerance, because argon grows from north to south of Morocco, and northern part and southern parts climate is completely different in terms of dryness. So the little bit analysis has been done for its 16 and ITS. We just did for fruits part. So from 38 sample, we were recovered around 139 ASVs from 41 different family members, 52 individual different genus. And for ITS, 24 family groups and 26 genus. And this is just the taxonomic, taxa abundance. And we can see for bacterial taxa, Erwiniaceae and Enterobacteriaceae are dominating. And in terms of fungus, Pleosporaceae and Cladosporaceae are dominating the fungal community. So we did not do the further analysis yet. This has been just done this morning. So we will be doing the further analysis, and there will be huge number of data sets is coming soon, because this is only from fruits. We also have root, rhizosphere, and also bulk soil, and also from different, geographical distributions. So another project we did at AGC in terms of metagenomic study is, some of you may know this plant is called Malva, and we did 16S and ITS studies because this is one of the plant distributing all over, I think in different continent as well, but in Morocco it's from dry to even normal region in northern part. And we found is a high microbial diversity, and in terms of bacteria, this was a non-leguminous plant, but we found a high abundance of rhizobium, even though this is a non-leguminous plant. And we also did ITS-based studies from the same plant, and there is a huge response of Alternaria and Fusarium species been found. And again, this is from the same plant, from same sampling region in all over Morocco. This has been recently published from our group in this year in microbial ecology. So beyond that, this is our part of our team, the whole AGC, our professors and scientists, and also staff, PhDs and postdocs. Also, I don't have all the new PhD students' photos. So this is the family of AGC, and we are contributing in a different way to, and most of us are working actually on microbiome data. So I hope in future the datathon will be really enriched, and I think we will be happy to do that. Thank you. Thank you for your time.